covering the rest of the course is just what I just went over. And we'll have one lecture, unfortunately only one, on grape insects. But I'll give you a good library reserve assignment for you that'll help you some on that. Okay, that's one thing out of the way. Next Monday, Dr. Alley asked me to announce this to you, that of course next Monday is a holiday, and what he would like to do is to have all of you have Monday lab come in either Tuesday or Wednesday. Is there anyone who cannot make either one of those days? All right, you have to come Saturday morning then, like you did last quarter, to pick that up. Because it's fairly important that you get that, and unfortunately school will be out, so you can't get it. What else was going to talk about? Oh, I've got these little reading assignment that you had. I mean, this little exam, Evolution of Vineyard, up here, which you can get after class if you want to pick yours up. The grades varied from 9 to 14. Only 2 people picked up the 14 points. I didn't grade it. I gave the reader some suggestions, and he's got down here the ones that he counted, the ones he counted double and single and so on. So if you really want to go over it, okay. But I think the main thing was that most of you picked up the lesson that I wanted you to get, and that is that don't believe everything you read. There's always a lot of errors in it. And somebody picked up, what, Frank? Is Frank here? Didn't you say somebody had 23? I think somebody had 23 errors in that little 3 or 4 page article. But it's still pretty well written, I think. Okay, now let's see if we can pick up where we left off last time. Can you hand those back? I'm caught. See if that's right. Yeah, here's that goes on top. And might as well hand these back. And that old one goes on top. The old one, that old set there goes second. We are going, the other reading assignment which you should have had was to cover phlox or nematodes, but that's in that chapter 18 anyway, I believe. So that's what we want to finish up this morning. You know, we got started on it last time. So let's see how far we can get on it today. Now, we started in with these slides, but because some of you weren't here and we rushed through them a little fast, let's just start and see them again here on the what phylloxera damage looks like and what the bug looks like. Now, this is the damage on the leaves. We'll discuss these leaf cycles and everything in a moment. Can you make that any darker at all? Oh, no, it doesn't make any difference. What we want to see are those bumps. The galls, as they're called. And you can see here, remember I tell you that the phylloxera, we don't have this form here in California, fortunately. This is the wonderful thing that's good about today's lecture, or about this pest, is that we do not have the flying form. So that if it's moved, it has to be moved as the aphid look-like thing. But this is in Europe and in eastern U.S. Remember, the female feeds on the top, makes a gall that hangs down below the leaf, and in that gall lays a number of eggs, which again hatch into other female laying egg forms, which can continue to spread. We'll go through the whole cycle here in a moment. I just want to let you see what the damage is. Okay, next slide. And here's what it looked like on the roots. Greatly magnified, but now you can get an idea why they call it an aphid, a root aphid. Normally, we like to have some of these set up so that you can actually see them on the wood under a mic, but the ones that we got and were carefully taken care of, I don't know what killed them off, and neither does Dr. Leiter or Ferrari, his technician, but we checked them out when I got ready to show them, and they've all died. I don't know why. So we were trying to cultivate them so you could set them up and see them, but we lost out this year. And one reason why we don't really dig them in this class I mean go out and dig them, is that in the overwintering form, and until about another month or so, they are very dark colored, just about the color of the bark. Here's some down in here that are really almost that. Well, you can see one in here. And they're very hard to see at this time of year, and it's very difficult to find them. But later on, when they begin to multiply rapidly, in warmer weather, they turn this nice golden orange color, and then you can pick up a root sometime where you have a nest of them like this, and you can see with the naked eye, even I can with my eyes, a yellow smear, like you'd smeared it a little bit with a little yellow makeup or something. And in a good bright light, you can see these, you can almost see the individual one, but just a little 5-power hand lens, you can pick them up and see them just about this good. So there they are feeding. You can see the mothers, and then the eggs that are being laid, and the larva stages halfway in between, and so on. It takes about, depending on how warm it is, anywhere from a week to a month to go from egg to mother, laying egg, mother, which again, in other words, to complete the cycle. Anywhere from a week to a month, depending on how warm the weather is, to complete this cycle. So you see they can multiply pretty fast because an individual mother can lay any great number of eggs. So they can multiply pretty fast in the right conditions. Okay, next slide. This is just, I'm just going through several slides here to give you a little more close-up. Now see, this is what they look like more about right now out in the vineyard. There's more of this difficult to see color. Next slide. And this is just, that wasn't as good as I thought it was. I thought it was a nicer close-up than that, just showing you these eggs and larvae again. Next slide, if there is no one. Now that's what they look like on the leaves and what the animal looks like itself. Here's the type of damage they do on the roots. And they feed, of course, on where they really do the worst damage is that they feed near the tips on these young roots, and you can recognize and distinguish the difference between phylloxerated rootlets and nematode rootlets by this little telltale characteristic, this little gooseneck type of thing that happens that you see right here near the end. Because wherever that phylloxera digs in, it causes this gooseneck bend, and the phylloxera is sitting right in here. So it really works over these young roots, but it also feeds on the older ones. These are about pencil size or finger size, and you can see the galls on the roots. Now you saw the galls on the leaves, but these are the places where they feed. And the phylloxera, and get this, the phylloxera is more damaging than nematodes, because although both of them are just sucking or feeding or parasitic insects, if you want to call them an insect, the nematode does not seem to inject any sort of toxin or anything into the rootlet, but the phylloxera seems to. Something like a mosquito bite. The mosquito feeds on you, but he also zaps you too with some something that makes it itch. And it's this toxin material, whatever it is, that really accelerates the decay of the little roots, or these decay areas. And that is what makes the phylloxera more damaging than nematodes on grapevine roots. But remember, we're going to talk about nematodes later this morning. Nematodes attack nearly every, well, not every, a great host of different types of plants. Whereas, as far as we know, phylloxera is specific for grapes, for vitis. Okay, next slide. And this is just a close-up of those galls. You see here, I just took a brush and brushed the bark off so that you could see where they fed and cause these sores, if you want to call that more vivid perhaps, on the roots. Okay, next slide. Okay, now let's stop here for a moment. You all received that drawing, but I thought a lot of you wouldn't bring it with you this morning, so we'll show it as a slide. But first we want to discuss a little bit more about this animal. I said the life cycle in California is simplified, fortunately. because all we have there is this matter of the entire population consists of females, and they produce asexually by laying these eggs. There's a great number of them. You can get 8 or 10 generations a year. And for some reason, I don't have a note here on how many eggs a female can lay, but it's a great number, of course, because they can have that, but apparently I don't. But anyway, I told you last time that phylloxera like heavy soil, and that they can't get along very well in real sandy soils. I told you something about the areas in California where they're present, in these heavy soils along the middle part of the San Joaquin Valley over on the east side, where they have some area of heavy soils. And I told you how that got in there to this other problem of bringing in rootstocks and so forth from the Napa Valley. Now, all of the north coast, where they grow grapes, is infested. And fortunately, at the moment at least, we don't know of any infestations in the Salinas Valley. And we just wish that there was some sort of strong enough, enforceable enough quarantine to make sure that the Salinas Valley can stay free from phylloxera, because so many of those vineyards down there are being planted on their own roots with these greenwood, these potted cuttings from the greenhouses and so on. And if we ever get phylloxera in there, it's good night, because it will spread pretty fast. Okay, so but it spreads even though we don't have the wing form. See, it was a wing form that went to Europe and just, in about 10 years, went over the whole country. Because, as we'll get here in the cycles in a moment, that's, of course, a real fast way to spread it. But in California, since we don't have the wing form, maybe I should have gone over that chart first, it's only spread by man, by spreading rootings that have phylloxera on them, or in very innocent ways almost, because it just takes a few phylloxera caught up in a few clods of dirt, spread in the vineyard, and then they really multiply in a hurry. And you can get this by, as I told somebody, and they've always been a little bit surprised, you can come over to my vineyard where I've got phylloxera and stomp around with your boots, and then get in the car and go home into your nice clean vineyard and get out there, and a few of those clods fall off that contain a few phylloxera, and you're inoculated. So you've got a good seeding there. Or fruit boxes that have a little mud in them. You throw those boxes, you've seen them throw them, they throw them out, and they get mud in the corners and so on. Or, of course, big tractor equipment with treads and tires and so on that move from one vineyard to the next. You may have contract work done for chop and brush or mechanical picking. Or any of these things can spread it pretty well. So the point is, and that we're asked this quite often, well, I live a half a mile from the nearest vineyard that might have phylloxera. Am I not safe? Well, you are if you have, like one guy had a place where he had a tub at a place where everybody walked in, had to walk through the tub of a disinfectant and all this business. So if you want to go to all this trouble and have a place where everybody comes in or drives from their vineyard to yours and so forth, you can do a pretty good job, I guess, of keeping it out. But in this general north coast area, unless you're isolated, see, like Salinas Valley, 50 miles from the nearest vineyard, something like that, then you better put them on rootstocks, because that's about the only way to control it. And we'll discuss rootstocks in a minute. But I think before we discuss rootstocks and control measures, let's see if we can go over this slide. I think it's pretty dim, but most of you have the handout, and hopefully some of you read it, so you've got a pretty good idea what it's all about. Here is the life cycle now as it exists in the eastern U.S. and in Europe. And the best way to start is to start down here on the roots, because that's what you're familiar with. That's what you just saw. So you have this female form that is laying all those eggs, and a great number of them, on the roots. And then sometime during the late summer, through some act of nature, a few of them become wing form and crawl up out of the soil. And this wing form is a female, and but this is a very short-lived or short-life female. She only lives for a few weeks. She doesn't even eat. She doesn't even have a mouth. But she lives long enough to lay 3 or 4 eggs. And these eggs are two types. A small one which hatches into a male, and a large one which hatches into females. And that's the only sexual part of this whole cycle on Phylloxera. Because this male and female then mate, and they don't eat either. They just mate. and then the female lays 1 egg on the bark or the trunk or in a crevice or something on the grapevine. Lays 1 egg. And then they kick off. They die. So these 2 stages here where this nymph or this flying form female comes out, lives just a few weeks and lays 3 or 4 eggs of 2 types, very short life, and then they mate, and then they only live a few weeks, and they don't eat, as say, and then the female lays 1 egg, and then she dies. And that egg overwinters. That's called a stem mother. And it's somewhere on the crack of the bark of the trunk or something. So you get 1 egg then that overwinters, and it hatches into a female, a stem mother. And then that stem mother still doesn't have any wings. So we only have 1 wing stage right here. But she crawls up out of that bark and so forth of the older wood of the vine up onto the new leaves and makes that pouch and starts laying eggs in that pouch on the leaf. And then as those eggs that are in that pouch hatch out, they crawl out, and then they move in a crawling form to other leaves and start the process all over again. And then some of those that are produced drop on the ground and crawl into the ground and get on the roots, and here we go again, seeing the cycle.
Do they live continuously on the leaves? I mean, all winter long or what?
Well, there are no leaves there in the winter, man.
Do they still survive?
No, they drop on the ground. If they're going to survive at all, they have to survive on the root system during the winter and on this system. So the only time you have them on the leaves, let's say, is when that early spring female egg hatches out, then she can crawl up on the leaves and it starts there. But the way it spreads so fast over Europe is this wing form crawling out and making these 2 forms which mate and then lay the egg. As I say, we have only this form in California. This root system right here. I don't know, or the part which is effective on European grapevines. Oh, on European grapevine. This is a Australian publication. What he means, the part which is affected on Vitis vinifera. It's a little bit confusing because this is the part that's really effective in California also, only this part here. But I think what he's getting at here is another little phenomenon, which is an odd one, and that is on species and varieties in which it really hits the leaves the way you saw it. It usually doesn't hurt the roots much. And where it really hits the roots, as it does on Vitis vinifera, it doesn't hurt the leaves much. Now, is that confusing enough for you? I can simplify a little bit. Most of our Vitis vinifera have a little pubescence or fuzz or hair or whatever you want to call it on the leaves. And this stem mother doesn't like that fuzzy leaf. But she likes these nice, slick, glazed rootstock leaves, or the wild vinifera like rupestris or AXR or 1613 or any of those that have a slick leaf, then that's where she really goes to town. So it's just an oddity that generally speaking, you have the reverse type of damage. The rootstocks now that you're going to use to get a phylloxera resistance, if we had where they have the wing form, they really get chewed up like you saw those, because those were rootstock leaves that you saw that were so puckered, because they were nice and slick and thin and really easy to feed on.
Is it just a climate deal that makes the wing form come out?
That question's been asked for 150 years, but we don't know the answer. I don't, the answer becomes even more confusing because they have the wing form in Mexico, and they have a dry climate, but they do have some summer rains. You know, what rains they get come in the summer, but it's not a lot of rain, 10 inches. But apparently there's something about that, because that's the only place where I've really seen them, and I never really believed they had them there until I actually broke open some of those galls with my fingernails and looked at the phylloxera in them. Well, no, because we don't have the wing form. But if we did, somebody has said occasionally they've seen a rare, nothing is absolutely constant in biology, in our biological world. So on a rare occasion, people have seen a wing form in California, but very, very fortunately it's sterile when they have found it. Or apparently it has to be, otherwise we'd eventually get some infestation. Okay, let's see. That takes care of that then, I guess. Okay. Well, I've got one more there I wanted to show. Let's catch that one because we're going to talk about control. And I've told some of you out in class that one way to get rid of them is drown the little beggars. And this is a French vineyard with a treatment in which they have a variety apparently with, well, I don't know whether this be a French hybrid or whether it's a vinifera. But under some situations, they can keep the phylloxera knocked down fairly well by just flooding the vineyard about every third or fourth winter for a couple of weeks. Remember when I was talking to you about soils and drainage and wet feet and so forth, I said the vine can stand wet feet while it's dormant. And I told you that that's one way that we do it, and I'd show you this slide later on. And that's southern France with an emergency control for phylloxera. But that's not a very good control. It's just sort of a get-by control. Or a couple of weeks at least. But I guess you could drown them. I don't know how long they can hold their breath. But the usual procedure is a couple of weeks. No. Okay, we have a couple of other controls. One control you can have, some of these references, so would give about 3 or 4 controls. Now don't write this down. Fumigation with methyl bromide and nicotine and oil and what have you and so on. But way back early when Dr. Leiter was first starting FPMS, or Ali was going to talk to you about it later on, the state required him to fumigate any material he sent out of here with methyl bromide. And that old refrigerator some of you have seen out at the field house was set up so he could fumigate small bundles. But it killed off phylloxera and nematodes on the rootings, but it also killed off the vines. So no matter what you read in the literature that methyl bromide is good, it isn't, because it does as much damage to the vines as the pest you're trying to kill. It's that old deal, the operation was successful, but the patient died. The only one, and this one to write down, the only one that you can use on nursery stock is a hot water treatment. And you might as well put in parentheses after this. This is also the treatment for rootings from nurseries suspected of having nematodes. And if you completely submerge these cuttings, or these rootings, or any cutting or rooting that you might think has phylloxera on it, you can submerge these for a period of 3 to 5 minutes in water at 125 to 130 degrees Fahrenheit. Everybody has his own little way of remembering things. 125 and 130. Somehow I get the 3 and 5 in there and remember that it's 3 to 5 minutes at that temperature. Because you can go on up to the next point, and 3 minutes at 130 will take care of them. Or 5 minutes at 125. So 3 at 130, or 5 minutes at 125. And what you've got to do is to have a very good piece of heating equipment. That tank that's about 4 feet square and about 4 feet high, and big, thick insulated walls that you see out at the back of our area there where you catch the bus, was built for that. I know Nelson uses it to make raisins, but it was built it was built as a fumigating tank because you've got to have very careful temperature control. See, we're talking about temperatures of 125 to 130, and very precise timing. So you have to have a good stirring equipment, good insulated tank, and very careful control. And then when you dip them, there's 3 to 5 minutes. You've got to dip them in relatively small bundles, and it's usually a good idea perhaps to pre-soak them in about 100 degrees or 110 so you don't do too much cooling of your water when you put them in. And then dump in there in small bundles. And then right out of there into cold water or into water to cool them off. If any of you ever blanched corn or something for a freezer, that's the process you go through. So because we're killing off the phylloxera, we don't want to kill off the vines, and 145 degrees temperature will kill the vines in 2 minutes. So if you don't have absolute good control and really go at it religiously and carefully, then you're going to lose more vines than you're going to save.
What about dry heat? Have you tried dry heat, like 24 hours?
I don't know why you couldn't use dry heat, because it seems to me that'd be easier to control. Or would it? But maybe it's a matter of penetrating or something. I don't know. That's a good question. I'll try to see what Leiter has to say about it. Leiter's our phylloxera man, and he used to give this lecture until he's teaching 9 hours a day, 5 days a week in plant science too. Okay, so that's about the only treatment you can use. You can use that French system of drowning them, or you can heat them this way. I think maybe part of the answer is that you're doing two things when you soak them this way. Maybe you're drowning them as well as cooking them. But of course, as you all know, the standard control is to use the rootstock. And we don't want to spend too much time on rootstocks because we mentioned them several times. But if any of you really want to get something about rootstocks, we do have this leaflet, which you can get up there in that extension office. It's grape rootstock varieties, and it's extension leaflet 47, or called AXT 47. And it gives the characteristics of these more important rootstocks that we use all the way through. We have St. George, AXR, 1202, 99R, and 5A, and SO4, and it covers the nematode rootstocks too, Harmony, Salt Creek, and all of those. And we're just going to touch on 2 or 3 here. We've talked about St. George all year long. It's a native from the Mississippi Valley where phylloxera is endemic, and therefore it has tolerance in the roots but not in the leaves. Of course, it doesn't do a whole lot of damage, I guess, on the leaves. It just looks bad, but it can really kill off the roots. So this stock is used. It's very vigorous, and some of you have heard me say a root, if you just throw it down on the ground, walk off and leave it. Almost. We used to throw away some trimmings, and the first thing you know, you'd see those growing. And it does quite well in dry soils. Its main fault is, of course, it's almost too vigorous. It causes the scions or the fruiting varieties to be too vigorous, and whether or not this is the direct cause or not, it causes straggly clusters and relatively poor set. It's a lazy rootstock, I say. Leiter and I have fun arguing about it. He still likes to defend it, and I tell him it isn't worth any of it, but because it make a big, vigorous vine and carry a light crop and straggly fruit. But it is our most resistant rootstock to phylloxera under our California conditions. Of course, the most, okay, then the next stock is AXR. And I don't want to go into a lot of the botany of these rootstocks in here because you get it 105. So I was going to put the parentage and so forth, but it's half rupestris. One parent of this is it's a vinifera, aramon. That's why it's called AR1. It's aramon, a fruiting variety of southern France, crossed with rupestris in some of that early breeding work that I talked about in 116a. And it's probably our best stock at the moment under certain conditions. In the irrigated soil, it isn't as drought resistant as St. George. And it is not as phylloxera resistant. It only has moderate plus resistant to phylloxera. But it fruits well. It has good vigor. Not as vigorous as St. George, but certainly better than average vigor. And gives very nice fruit. But remember that you've got to have it won't bear up under these extreme conditions of very heavy phylloxera infestation or very dry, shallow soil situations. But since most of you are going to be using sprinkler irrigation and so forth from now on, I think that this, again, is a kick against St. George. Why plant St. George if you have water available to give just one good irrigation? Now, one thing about both of those stocks which is bad is that they have no nematode resistance. So if you have a situation like Lodi and some of these intermediate moderate soils, and you may have troubles with both phylloxera and nematodes, you're sort of stuck. What do you do then? Well, the best compromised general purpose stock so far that we have probably is 1613. And it does have its problems, but it has very high resistance to root knot nematode, which is our most common one we'll talk about in a moment on grapes, and has moderate resistance, not as high, I guess, as AXR, but sufficient under some of these mildly infested situations where you have a sort of a sandy loam soil and the phylloxera have a little trouble getting by anyway. But once again, this stock has a little problems. Sometimes there's a little problem with late ripening. And with colored grapes grafted onto 1613, sometimes you have poor color, quite noticeably poor. In some of the trials I'm carrying on, we can put down the various lots of Cardinal, for example, or Emperor, and you don't have any trouble picking out the one that came off 1613. So we do have some other stocks that are coming along with a little better resistance, a little better possibilities. One of the most recent ones is one called Harmony. And Harmony has shown greater resistance to both nematodes and phylloxera than 1613. A little better resistant to both of them than 1613, and a little more vigorous fruiting varieties on top. So it's coming along and becoming a very popular substitute or replacement for 1613. And then we have one that is just hitting the market now, if I can find it here. I guess I haven't got in there. Does anybody remember what this new one that's just out is called? No, it's a sister or a back hybrid, I guess, with Harmony. Freedom, yeah. Freedom is the latest one out. Thank you. And both Harmony and Freedom were developed by Mr. Snyder, Dr. Snyder, at Fresno. And that was the Fresno, USDA Fresno grape station. And the two men that worked there for years and years as a team were Snyder and Harmon. So he did Harmon an honor by naming this Harmony stock after him, just as a little aside. Okay, that then covers the phylloxera picture, except that surely there's some questions. Anybody got questions? We got a couple of minutes. Yes? I don't know. I don't like liver. Maybe some of these rootstocks just don't like the taste, or phylloxera don't like the taste of these rootstocks. I don't know. We're lucky.
The nursery, is this after they grow for a season in the nursery that
What's this now?
What do you use the hot water treatment?
Well, you see, if sometime you goof up and plant a nursery in a place someplace, and it happened to be infested with phylloxera. Well, I'm not going to buy your rootstocks with the possibility that there's some phylloxera on them, because there might be a few that his question then may confuse here. I don't know. There must be a few of these phylloxera that will get on those roots. I don't know that any of them are absolutely immune. But even if they weren't, you'd still, if you add that soil there, you can have residual old roots from an old vineyard or something that contain these phylloxera that can keep the population there for some time. And then you get dirt on these rootings, and then transfer it to somewhere else, and you don't want to do that. Yep.
Is there a way to discover and control phylloxera that's been part of their life cycle?
No, you see, we're going to talk about nematodes next time, and we've got systemics, nemagon, DBCP or something, and that works fairly well on that, on nematodes, and there's quite a bit. There's a continuing program, but just at a sort of a low rate of speed, a low input, trying to find a systemic for phylloxera. But even this fumigation, which we're going to talk about, the phylloxera can get spread so easy and can build up so fast after that you cannot count on this methyl bromide fumigation and so forth to knock your phylloxera out so that you can use own roots. Now, most vines can take nematodes a little better. And if you knock out most of the population with fumigation, or if you didn't have them in there to start with by some other crop, and then you go in and plant these own-rooted vines in these nematode areas, and then baby them. Give them plenty of water, maybe even give them fertilizer, God forbid. But fertilize them, give them plenty of water, and crop them lightly by doing a little thinning until they're established, 5 years old or so. Then most of them can sort of take care of themselves from then on. You probably get 3 or 4 or 5% drop in crop, which I sort of object to. But Dr. Leiter insists that over the long run, conditions of that type are probably debatable, whether to spend the money for fumigation, which isn't going to last anyway. They're still going to come back in 5, 10 years. Whether or not you're better off just to forget it and put them in and really try to take care of them for those first 5 years and then live with the situation. You had a question?
How long will phylloxera survive in the soil after it's been pulled out if it's heavily infected?
Well, they should survive as long as there's any live roots at all. But I don't know how long they could survive. They don't form a cyst form, you know. Some of these nematodes form what we refer to as a cyst. It's sort of goes into its shell. And some nematodes can last, I don't know, 5 or 10 years. But phylloxera is a soft aphid type thing, and it should not survive beyond the presence of any live roots. But you can have live roots down, 8 or 10 feet, and it's pretty hard to kill them. Apparently not. Apparently they can go just as far as the roots go. So, on the other hand, can they? Because if you can drown them, they must have to have air. All right, that's one thing we find out too.
Don't they grow on the rootstock, so you just don't do as much damage to them as on root?
Well, that's a question I'm not sure of whether or not you don't find a lot of them on roots. If they exist on roots, they're in very, very small numbers. It may be just like an animal eating grape stalks. If it starves bad enough, it may decide to eat them, I think. But I'll pick a couple of these points from Dr. Leiter and bring them back next time. Okay, these old exams up here, if you want to get them, I think it's sort of nice to keep that.